with reference to the northeast Indian snakes. Because over the years we have been working with some of the medically important snakes of this region. So first of all, why do we need to understand, or why do we study venom, which we basically call it as a venomics? Because when we try to understand the various components that is present in the snake venom, what it gives us a profile of the pharmacological effect these proteins can have. And as Professor Sakti told, that they are very precise in exhibiting their activities. That's how we try to get to know the mechanism of actions. And using this kind of understanding, what we can use is we use it for developing therapeutic molecules. And the more importantly, it's understanding the venom profile will help us to improve the efficacy of the Indian antivenoms. And another lacuna that we have in India is that the lack of what you call the diagnostic kits. So it's through the understanding of the various venom components that is present in the snake venom, that's how it will help us to develop a rapid diagnostic tool, which is the need of the hour for proper management, which will help the clinicians to deal with the snake bite patients. And already Professor Sakti has already described how these proteins have been used to develop drugs. But still, I would like to focus on what exactly is a venom. Actually, venom is a complex mixture of proteins and polypeptides. And remember that these are what you call an evolutionary adaptation in the snake venom, just to overcome selective pressures they have from the prey as well as the predators. So there's an adaptive evolution in these organisms. And if you look at they contain around from 30 to several hundreds of venom proteins, and these venom proteins, they differ as per their what you call the location or the habitat is concerned. For example, if you look at the land snakes, they are more complex as compared to your snakes that live in the aquatic environment. And these proteins and polypeptides, based on the structure and functions, they can be classified into enzymatic as well as non-enzymatic, which I don't want to go in detail already. Professor Sakti has elaborated on that. But what is important for us to know is that there is variation in the venom composition, and this fact is already well documented in the literature. And this could be within the species, among the species, within the genus, among all. It may be different within a single species in the population. There could be differences of venom composition between male and a female. Even there is ontogenic variations in the venom compositions. And as per these reports of Doley et al. Way back in 1996, they have clearly established that the main driving force of this variation is basically the prey availability. And what is the importance or the consequence of understanding this venom variation? Number one, what we will understand the venom toxicity, which is due to variation in the venom composition. This will lead us to understand the pathological symptoms that this venom can induce, and most importantly, the inefficacy of these antivenoms, as well as it will help or which has an impact on the primary venom research. And we have also seen that even in the venom composition between the mother and the siblings, or here if you see there are three panels. One is the panel that is the analysis of the venom of the mother and the first class as well as the second class. If you look at here, it seems there is no variation in the illusion profile at all. But if you look very closely, you will find that there are minor variation in these, and these variations are because of the presence of different isoforms. So because of these, it can also affect the neutralization efficacy of these antivenoms. And recently what we have seen is that even within the male and female, that if you focus in these two bars, there is differences in the height of this bar. So there is differences, subtle differences even within the venom composition of the male and female. Here it is in case of the Naja kaouthia that we have analyzed from Mizoram. So but when we started working with snake venom in Assam, we started first with the Naja kaouthia venom, and we were fortunate to have samples from different regions, and the main objective was to see whether there is any variation in the Naja kaouthia venom of eastern India. So simply we did an SDA space. You can see there is differences in the vein, presence or absence of the vein. This clearly tells that there is variation in the venom composition, and this is more clear when we do a reverse phase HPLC or separation of the venom proteins. So if you see, we have divided these venom HPLC profile into different regions so that it becomes easier for us to understand. If you look at region 2, in case of the Assam venom from the Assam, there is a clear distinct peak which is absent in the other venoms from different other regions. So region specific variation of venom is clearly established even with the Naja kaouthia venom from the Assam. So now what does it do? What is the impact? So as we already been discussing that these polyvalent Indian polyvalent antivenom which is raised against Big 4 is used for treating Naja kaouthia. So this is a Western blot analysis, the panel if you see where I have highlighted with red box, that some of these bands are not recognized. So if there are antibodies against these venom proteins, then definitely this could have been bands or this could have been recognized. But since the antivenoms are raised against Naja, not Naja kaouthia, so treating Naja kaouthia patient with these polyvalent antivenom, definitely you will be requiring more vials of antivenom, which actually will lead to the secondary complications that we know already, the limitations of the polyvalent antivenom. But if you really want to understand the variation in the venom composition, it is important for us to understand the whole proteome of the venom. So whole proteome means the every protein families or the proteins that is present in the venom has to be identified. So this we have done for the Naja kaouthia from Assam using chromatographic approach. First these venom samples were separated with reverse phase, then each of these peaks were analyzed using mass spec. So based on the mass spec analysis, this is how the various proteins have been identified. And if you look at the overall proteome profile of these snake, what you find is that 58% of these proteins are neurotoxins. As you know, neurotoxins, they will be targeting the neuromuscular system of a victim and a prey. And overall, in the Naja kaouthia venom of Assam, what we found is that there are about 69 different proteins which we can classify into different 9 different protein families. So we want to see how it is similar or dissimilar with other Naja kaouthia venom from Southeast Asia. So if you see in the pie diagram, you can see here the one with which the red box is the one that is from the Assam sample. So here, if you see 87% of the venom protein is neurotoxin. That's why the immediate signs and symptoms that you see of the Naja kaouthia victims will be always neurotoxicity. So, but the story doesn't end there, but we wanted to identify what are these proteins in the low molecular weight protein that which is not recognized by the polyvalent antivenom. So using a technique called next generation antivenomics, I'm not going again into the details of the technicalities of these 3G antivenomics, but simply remember this is a pull down assay using the polyvalent antivenom. That means those protein that is present in the venom is recognized, will be removed, and those that are not recognized will be retaining in there. So these boxes are the one which is not retained. So, and we wanted to identify exactly what these proteins are. So if you see in the previous slide, I showed that the polyvalent antivenom does not recognize the low molecular weight proteins. And also these, through the 3G antivenomics, we could identify that actually it is a long neurotoxin. So long neurotoxin again is a member of a family of protein called the three-finger toxin, and this particular protein which is not recognized by the polyvalent antivenom present in Naja kaouthia is actually alpha cobrotoxin. Now what is this? This is a presynaptic neurotoxins, and it blocks these acetylcholine receptors leading to muscle paralysis. So even if you are treating these kind of victims with polyvalent antivenom, this effect is not going to be subsiding very easily. But since these Naja kaouthia belongs to the same genus of Naja, so we wanted to see how similar and how they are different. So red profile is that of Naja kaouthia, the black one is of Naja. Here you can see there is presence or absence or difference, and if I magnify a particular area, you can see this particular one is that one it is not present in the Naja, and we know that Naja is used for production of the polyvalent antivenom. The another now medically important snakes of these what Surojit was mentioning is these green pit vipers. So we also had the opportunity to collaborate with Dr. Giri and to see the pharmacological profile as well as these venomics of these particular snakes. So, as he was mentioning that of late, between 2018 to 2022, there were 92 patients which was bitten by this green pit biter, either salazar or erycturus. But the recent is unpublished data from his hospital, what we see is that this is the one that is because of the green pit viper. So this again, because of this awareness as well as those reporting to the hospitals, they could clearly see that these green pit vipers are more prevalent in those in the upper Assam part of the state. And from his hospital record, what we found is that if a person is bitten by this green pit viper, they have to stay longer period of time in the hospitals. And as he was mentioning that then these antivenoms are not working, only they have to do a conservative treatment and observe them for a long period of time. But what triggered us is that why does a patient has to stay long in a hospital when it is bitten by a green pit viper? That's when we found that a particular protein called, now we are naming it as a erythrofibrase, which is uniquely present in the what you call the green pit viper, and these are a protein which actually disintegrates or degenerates this alpha vein of this fibrinogen. So imagine the person is bitten by this green pit viper and immediately it mixes up with blood. So what it is doing is it is degrading the alpha vein of the fibrinogen. So if these veins are not there, then what we can see that the proper blood coagulation will not be there. That's the reason that they have to stay for a long period of time. So these are some of the biochemical assays by which have confirmed that this is the protein that is there, which is responsible for non-coagulation of the blood and prolonged stay in the hospital. And we also try to see the variation of this venom composition with different other green pit viper from Himachal, Mizoram, and from other Arunachal. And what we can see that this is an SDS profile by which you can see the presence or absence of bed. So this clearly tells us that there is variation in the venom composition. And again we have clearly shown with the HPLC profiling that there is differences in the elution pattern of the venom proteins present in this green pit viper snake. So there is variations. So now we wanted to see that how these polyvalent antivenom reacts with these. So if you look here, what we have is a monovalent antivenom, that is GP, VAV, it is a green pit viper antivenom. The other one is PAV, is a polyvalent antivenom. So polyvalent antivenom is that of an Indian origin. You can see here these boxes do not have any band. That means this polyvalent antivenom does not recognize any of the bands present in that. But polyvalent antivenom that is raised against green pit viper could recognize all these. That's where what we require is a specific antivenoms for treating specific snakes. So again, this also we have confirmed with these antivenomics approach, where you can find that there are many proteins which is not immunoprecipitated by the polyvalent antivenom. So again, to further understand the variation in that, what again we have done is that we have used these chromatographic approach as well as mass spectrometric analysis to see what are the venom proteins present there, and that's how we come up with the proteome profile of this green pit viper, specifically Trimeresurus erycturus, where we found that there are 53 protein which belongs to 10 protein families. Now, and if you look at and compare with the proteomics of the Southeast Asian, this is how it looks. And here, just to show you that here we the Trimeresurus erycturus from Mizoram, if you see that serine protease is the most abundant one here. Also, where you find that there are a lot of phospholipase A2 enzyme. So I'll just quickly rush with this. Very common again, what you call this Bungarus fasciatus. Again, we had the opportunity to have venom samples from West Bengal, Mizoram, as well as from Assam. Here also what we know is that Caerullius is used for production of the antivenom, not the what you call Bungarus fasciatus, but still the polyvalent antivenom is used. So if you just compare the elution profile of the venom proteins, this is how it will look. The red one is that is of the fasciatus, which is very different. It tells you that there is variation in the venom composition. Again, not only that, the other venom of Bungarus fasciatus from these locations were also compared, and if you look at the close comparison, you can see the presence or absence of venom. Same snakes, but from different geographical location, they show differences in the venom proteome. And when you look at the cross-reactivity with the polyvalent antivenom, you can see here these box where there in the SDS space there are a lot of bands, but in case of the Western blotting, you can see that all the bands are not present, which tells you that there is absence of particular antibodies in the polyvalent antivenom. Again, we have used these antivenomics to identify that protein. One interesting observation we found is that actually there is a phospholipase A2 protein that is present in fasciatus, which is different from the phospholipase A2 enzyme from the big 4 venoms. So now everybody knows what is big 4. They also, all these, any snake venom, what you will find is that phospholipase A2 enzyme will be present. But here, in case of the fasciatus, this particular phospholipase A is sequentially different from the other phospholipase that is present in the big 4 venom. That's why in the dendrogram you can see it is branching out separately. That means again this is not recognized by the polyvalent antivenom. So these are our observations. Today we are quite delighted that the forest department is agreeing to have a venom collection center, which we think that the shortcomings that we have in venom research will be taken care. And because what we want is that we want a region specific antivenom. So based on our understanding of the venom and antivenom cross reactivity, what we have realized that the available polyvalent antivenom is not the solution for treatment of snakebite patient in Assam. And having a venom center here collecting or having a region specific venom collection facilities will help us to understand the biochemistry of that cross reactivity, and also will help in developing what we call the region specific antivenoms. So before I end my presentation, I would like to check my funding agency, Tezpur University, Department of Biotechnology, ICMR, as well as the DST, and also my PhD student Archana, Susmita and Amit, whose work I have presented here. Of course, these are the list of my well-wishers and collaborators, without which I would not have been standing here in front of you. Thank you very much for your patience.